Hello, my name is Peter, and today I will be presenting our work on if tissue source sites leave identifiable signatures in whole slide images beyond staining. Computational pathology is an emerging field that aims to leverage the power of machine learning in order to aid doctors, for example, by improving diagnosis accuracy. This means the field has a strong potential to save many lives. However, when we make use of machine learning, we have to ensure any model we design can be seen as trustworthy, which can only be achieved if our models are robust and have good generalizability to unseen data. In this work, we focus on models that make use of image data, known as whole slide images. These images are created from patient tissue samples that have been stained in order to highlight important features of the tissue and then scanned. On the right, you can see one such whole slide image for breast tissue. One of the main problems of using these images is that they are highly susceptible to domain shift. Some examples of this include the duration of staining, how much staining agent is used, the scanner used, and potential of various artifacts such as dust, hairs, pen markings, and so on. This means that many computational pathology machine learning algorithms often adapt poorly to a clinical setting by seeing a significant drop in performance compared to the lab. As such, doctors may not be willing to use these models as they are unreliable. Currently, the main adopted solution in the field to this problem is stain normalization. The idea behind this is to try and transfer the color distribution of a target image to other images in the dataset while preserving tissue information. There are many stain normalization algorithms that are currently used in the field that to the human eye seem to solve the domain shift problem. This can be seen above, where the patches look similar after Marchenko stain normalization. Nevertheless, even with stain normalization, many models still fail when they are deployed in a clinical setting. We argue that this is because stain normalization only works on low-level features, meaning signatures left from labs in higher dimensions are not accounted for. This means many current models cannot be seen as trustworthy as they cannot generalize well to unseen data. The debate of if stain normalization is helpful is a widely discussed topic, with various papers reporting results supportive of both sides. For example, Salvi et al. Showed that for prostate tissue, the application of stain color adaptive normalization seemed to increase accuracy by a substantial amount, from 0.7 to 0.79 on a test set. However, the work of Srufi et al. Showed that Marchenko normalization didn't help with improving accuracy of popular CNNs such as MobileNet, with stain normalization even doing worse in terms of accuracy when using VGG16 architecture. As such, the debate of if stain normalization is helpful in solving domain shift is still ongoing. In previous work, the study of site-specific digital histopathology signatures was researched with a different and independent experimental setup. Similarly to us, this study found that it is possible to easily identify site digital signatures using deep learning. In contrast to us, this paper made use of patch-level predictions, whereas we focused on slide-level predictions. It is important to note that our work is independently inspired and we were not aware of this paper's existence before submitting our paper. In this work, we wanted to create a carefully controlled experiment in order to measure the effect of stain normalization. To do this, we designed a simple experiment in which we predict the site of origin of a whole slide image, both before and after stain normalization. This was modeled as a weakly supervised binary classification problem, where for each possible site, we use a 1v rest approach. The fundamental principle behind this experiment is that if stain normalization is an effective strategy to remove an identifiable signature of the site of origin or the underlying batch effect, we should get substantial decrease in accuracy of predicting the center after stain normalization. For this experiment, we used two predictive models, which fundamentally work differently in order to marginalize the effect of choice. The first model is clustering constrained attention multiple instant learning, also known as CLAM. This is a popular method that considers each whole slide image as a bag of patches and then uses attention-based pooling function for obtaining slide level representations from patch level representations. For the second predictive model, we used MMD kernels. These are a new form of support vector machine-based classification that construct a whole slide level kernel matrix using maximum mean discrepancy over shufflenet derived features representations of patches in whole slide images. The important thing to notice here is that these two methods fundamentally work very differently. This means we can marginalize the effect of choice as our results are not specific to one type of method. For our dataset, we used 1,113 publicly available breast cancer whole slide images from the Cancer Genome Atlas program. These originated from 49 different source sites. However, we only predicted the source sites which had 50 or more images each, leaving us only 8 sites. To train and test our model, we used 5-fold stratified cross-validation, where 30% of the training data was reserved as a validation set for hyperparameter selection. First, we look at the results of CLAM. Here we can see that before stain normalization, the predictability of a source site is very high, with an average AUC-ROC of 0.94 across sites, indicating that source sites do leave signatures that can be picked up by machine learning models. However, even after the application of 2 different stain normalization techniques, Reinhardt and Machenko, the AUC-ROC was still very high. This suggests that stain normalization does not solve domain shift, as if it did, the AUC-ROC would have been near random, around 0.5. Next, we look at the results of using MMD kernels. Here again, we can see that before stain normalization, the AUC-ROC is very high, 0.98, and there is not a significant decrease in AUC-ROC after stain normalization. This again highlights that stain normalization is not an adequate solution to the domain shift problem. After using MMD kernels, we also visualize the generated kernels. Here, a yellow-red color indicates greater similarity between whole slide images. From the diagrams above, we can see that both non-normalized and stain-normalized whole slide images have a large proportion of brightly colored regions in the heat maps, indicating that there are many slides that share similar characteristics. In conclusion, our work has provided empirical evidence for the existence of source site signatures. This is significant as it demonstrates that whole slide images contain more severe types of domain shifts beyond what can be handled by stain normalization methods. Of course, stain normalization and estimation methods may be useful for other tasks such as stain quantification, which is determining how much stain there is. Stain normalization will not be helpful in domain generalization, as it does not remove site-specific information from whole slide images. We urge pathologists to keep this in mind, that stain normalization may not be enough when designing experiments, and as such, we will need better approaches to handle domain shift. One such improvement may be to use domain adversarial training with gradient reversal, which tries to take into account the domain shift present in datasets. Thank you for listening.